7.37. Explain the four levels of protein structure, explaining significance of each level. We saw this in 2 point something, 2.34 or something. So way back in biochemistry, we talked about this. So it's review. So there are four levels. The four levels are primary structure, secondary structure, tertiary, which means 3, and quaternary, which means 4. So each one of these levels, they want you to talk about bonding, like what kind of bonds are going on to keep them in that shape, and just generally what the shape is. So real quick, primary structure is simply the order. Who's first? Who's second? Who's third? Who's first? Oh, methionine. Who's second? Oh, valine. Who's third? Serine. Who's fourth? Oh, valine again. So it's just what are the order of the amino acids, and very often we just draw them in like a chain of different colored circles. So methionine and valine and so on, right? They kind of did that down here as well. The bond that holds them together. What is the bond that holds two amino acids together, I ask you? Peptide bond. Yes, so I got that right down here. So that would be the bond that you would need to know. Secondary structure is seen mostly in fibrous proteins. Not always, but quite often in fibrous proteins like hair and nails and things that require support. And there are two types of secondary structures we need to know. One is the little twirly thing, and if I kind of wish it was a little better, but it looks kind of like a double helix that we see in DNA. So it has this like curly thing. And the bonds that hold this together are hydrogen bonds between the NH group of one amino acid and that CO of the other, near that carboxylic group. And if you can imagine, maybe the NH is there and the CO is there, and between them, we have our little hydrogen bond. The second type of secondary structure we need to know is something called a beta pleated sheet. You get it right up there. So our alpha helix was the first one, our beta pleated sheet. They often draw it as an arrow, but it really has like this folded structure. And there's interactions going on between them as well, forming these hydrogen bonds between the same groups of atoms. Tertiary structure, the next one. Tertiary structure is the most complicated because there's really four things that are happening to form this. So the tertiary structure we find in globular proteins. These are like enzymes and hormones that have this three-dimensional shape. But there are amino acids that are hydrophilic. I love water. That's what they say. And we see them on the outside of the protein because they're hydrophilic. We also have hydrophobic ones, and we would find the hydrophobic ones on the inside. So those are two of the four characteristics that we might find in a tertiary structure. The third one is that you see like these ionic bonds between acids and bases. So opposite charges attract. So maybe we have like here we have a positive amino acid, and then down here we have a negative amino acid, and their opposite charges form these ionic bonds. And lastly, there's this weird one called the disulfide bridges. And that is formed by two molecules of cysteine, which form, which will form a covalent bond. So they'll form a covalent bond between them. So if you can imagine a cysteine here and a cysteine here, those two would connect, forming our protein to screech this way, come down this way, so they come together. And quite simply, the last one, quaternary structure, all it is two basic things. First, it's multiple chains of polypeptides. So here, we can see that this one goes there. And we have a third yellow, I'm sorry, can't count, a second yellow one, a third green one, and a fourth pink one, all separate, but together they create one whole functional protein. And they're held together by either hydrogen bonds, or sometimes they're held together by these sulfur bridges. So that is the four levels of protein structure. And because of that, they will behave quite weird. No, not weird. predictable um, as they form, as they interact with membranes. So if you're looking at this structure, if this green part is a chain of amino acids, and again, I just don't like it when they just draw it as globs, but if you can think of amino acids here, all these red amino acids that I'm drawing are near the water because they're on the outside of the cell. It's watery there. So they will gravitate towards the outside. And here, inside, I have some green ones. These amino acids are like, I am hydrophobic. I hate water. But you know who I like? I like my hydrophobic phospholipid tails. They're my buddies. So those you'll find inside the phospholipid bilayer by those lipid tails. And that's it for protein structure, the 4 levels and how they will interact when we're talking about them in membranes.